Alrighty, everybody. So we're going to do a lecture midterm review. Alright, so remember physiology is going to be tightly connected to anatomy. Anatomy is the structures, but physiology is going to be the study of normal functioning of a body, right? If it's abnormal functioning, then it would be patho, right? But you gotta remember this understanding of structure and function, how they're closely related, because your structure will determine your function, right? So protein structure will determine how that protein will function and how it will respond in its environment, right? So remember, structure determines function. Sorry, let me go back. And so we talked about a lot in physiology, homeostasis. Homeostasis is basically maintaining a relatively stable internal environment. Homeo means similar, stasis means stable, right? So we're trying to maintain a stable environment. And if we're going out of that, range, right? For example, temperature, if you're going out of that range, we want to bring it back within this normal range and thus maintaining homeostasis. And so it's those negative feedback loops that are homeostatic, right? They stabilize a variable, they bring you back to homeostasis. And if you fail to maintain homeostasis, then you're in a disease state, right? Or a pathological condition, okay? And so when we're looking at the body's internal environment, we know that the extracellular fluid is going to be the fluid surrounding the cell. The intracellular fluid is going to be within the cell, right? The fluid in the cell. So the extracellular fluid, that's going to be the interstitial fluid, as well as plasma, right? And again, intracellular fluid is going to be the fluid within the cell. Now, it's this extracellular fluid that's going to serve as a buffer zone between the outside world and the intracellular fluid, right? And so that extracellular fluid needs to be kept relatively stable. So you also got to remember these ions and where they are abundantly, right? So intracellularly, inside the cell, you're going to find more potassium. However, outside the cell, in the extracellular fluid, you're going to find more sodium and, of course, more chloride, okay? Just some physiological concepts to remember. We're going to be carbon beings, right? We're made up of mostly carbon. Most of our weight is going to come from a carbon atom, okay? We know that our body is going to be made up of the basic unit of life, which is going to be a cell, right? And these cells, our cells, are going to be surrounded by a semi-permeable phospholipid bilayer membrane, okay? I talk about it a lot. Remember these four major biomolecules: carbohydrates, lipids, proteins, nucleic acids, right? Carbohydrates are going to be made up of monomers, right? Lipids are going to be made up of fatty acids and like a glycerol, right? Proteins are going to be made up of repeating units of amino acids, right? Nucleic acids are going to be made up of repeating units of nucleotides. But remember, the four major biomolecules, again, are carbohydrates, check, lipids, check, proteins, and nucleic acids, right? Remember that the mitochondria will produce the ATP for the cell, right? That energy currency for the cell, right? And ribosomes, whenever you think about ribosomes, you always want to think about protein synthesis, right? And so those ribosomes, they can be free floating in the cell, or they can also be studied on that rough endoplasmic reticulum, right? That's what makes it rough. And again, the rough endoplasmic reticulum is just a set of hollow tubes, a membranous structure of hollow tubes that's continuous with that nuclear envelope. But this is where we're able to make proteins at. Now, we also have a smooth portion of the ER, the smooth ER, and that's going to be important for lipid synthesis. Remember that, okay? Remember, like I mentioned before, these negative feedback loops, they try to restore homeostasis, right? So if, for example, you're hot, your body will try to cool down. You're thirsty, right? Your body will seek out fluid, right? Remember those things. And again, in positive feedback loops, they're going to push you further away from homeostasis, right? So such as childbirth, or the release of oxytocin, the stretch receptors and things like that, as well as baby suckle at the nipple. Again, you're pushing further from homeostasis, okay? Energy is simply used to perform work. Energy is the capacity to do work. And so we have different types of energy. Mechanical energy, like contracting your muscles. Transport energy, moving things, is transporting things. Remember chemical energy. Chemical work, right? Chemical work is the making and breaking of chemical bonds, okay? when we're looking at thermodynamics, right? Thermodynamics is simply the study of how energy is used. The first law of thermodynamics is that law of conservation of energy. It states that the total amount of energy in the universe is constant, whereas the second law of thermodynamics is that law of disorder, basically the law of entropy, basically saying everything in the universe tends toward randomness or disorder. Disorder is also called entropy. Enzymes are types of proteins. Proteins do everything in the body, right? Enzymes will speed up the rate of a chemical reaction, right? Enzymes are called catalysts. Again, these catalysts speed up the rate of a chemical reaction. How? Because they lower the activation energy, or simply the energy needed to start a reaction. Okay? Remember that, that's important. Enzymes are catalysts that speed up a reaction, right? By simply lowering the activation energy. The reactants that an enzyme act on is called substrates. And so when an enzyme works on a substrate, right, that enzyme is unchanged. It's able to make or break covalent bonds in a controlled way, and that enzyme can then leave that reaction unchanged and can do the reaction over and over and over. Okay? And so some enzymes, they actually need cofactors, right? Some proteins need cofactors. These cofactors are non-protein chemical compounds or metallic ions that are required for an enzyme to be functional. So things could be like B vitamins like folic acid, coenzyme, magnesium, calcium, zinc. These are cofactors needed to bind to an enzyme to make it functional. Okay. Again, as you can see, this cofactor, it binds to this inactive protein, thus making it active. Okay. And again, a protein or an enzyme will bind, or a ligand will bind to a protein. But notice how it's inactive right here, right? Something needs to bind to it, right? To activate it. And now it's active. Okay. Some things can bind and won't activate it, right? In a sense. So we have inhibitors. We have allosteric inhibitors. So an allosteric inhibitor will bind to an allosteric site. An allosteric site is any site that's not the binding site, or that's not the active site, right? So if something binds there, like an inhibitor, it binds to somewhere on the enzyme, right? But it renders it inactive. Notice how right here, the active site looks like this, but when this allosteric inhibitor binds, notice how the active site changes shape, thus not allowing for this ligand to bind there, right? It can't bind. So an allosteric inhibitor binds to somewhere else on the enzyme, changes that active binding site shape. Now that original ligand or that original substrate can't bind there no more. A competitive inhibitor binds at the active site. Right? It competes for the active site, and now the original ligands can't bind there. Okay, there are multiple factors that affect a protein's affinity or a protein's binding. Temperature, pH, right? So if the temperature is too high, right, or too low, right, that will affect how this protein will act, basically, and it probably won't bind, right? If it's out of its original pH, right, it won't bind, right? So these are factors that will affect how a protein will bind. How much of the ligand is there, right? How much of the enzyme is there? Again, these are factors that affect protein activity, okay? Again, how much of the enzyme, how much of the substrate is there, the temperature. These are things that will affect how an enzyme will bind. But again, some enzymes, they need to be activated, right? So you'll have those coenzymes such as vitamins, ions, things like that, that needs to bind to the enzyme to make it active. And again, remember, they're only will respond to their environment. They'll only work in certain conditions, right? In certain temperatures and certain pHs, okay? And again, enzymes lower the activation energy of reactions. So when we're looking at metabolism, metabolism is simply defined as all the chemical reactions that take place in an organism, right? And so we have catabolic or catabolism, is where we're breaking things down anabolism is where we're synthesizing things, and I'll talk about that a little bit more. And so metabolism such as glucose, right? When we're making ATP, we can make ATP, that energy currency, we can break down glucose, we can break down proteins, and we can break down lipids. We can make ATP from all these things, such as glucose, proteins, and lipids, right? When we break down glucose, we're going to make pyruvate, and we'll talk about that. Let me go back for a second, right? So glycolysis, the breakdown of glucose into pyruvate. Glycolysis will happen in the cytosol. This is an anaerobic process. We don't require oxygen. Again, this happens in the cytosol. Okay, when we are making that pyruvic acid or that pyruvate then gets converted into acetyl-CoA, and it's the acetyl-CoA, it's the carbons from the acetyl-CoA that go into the citric acid cycle. Remember that. And so the citric acid cycle is going to occur in the mitochondria. Okay, in the citric acid cycle, we're going to be creating a lot of these FADH and these NADH molecules. Why? Because these are electron carriers. We're going to carry these electrons to the electron transport chain, right? And when we drop these electrons off, these electrons will go through these different proteins, right? Ultimately, it's oxygen. Oxygen is going to be the final electron acceptor, okay? And it's going to be able to combine with hydrogen to form water ultimately. Okay? When we're looking at proteins do everything in the body. I told you that, right? But they're going to be created from DNA, right? So remember that central dogma where this is that central dogma where DNA is transcribed into RNA, and then RNA is then translated into protein. Okay? Remember the basis. In DNA, we have adenine binding with thymine. Remember that base pairing. Okay? You have to remember. Adenine binds with thymine. Guanine binds with cytosine. Okay? ATGC. In RNA, adenine binds with uracil. Guanine still binds with cytosine. Okay? And these bases, these nucleotides, right? They provide energy and they're part of that genetic material. Okay? Remember that. And so, remember, right? Central dogma again, DNA to protein translation. Okay? I'm sorry, DNA to RNA transcription, RNA to protein translation. Okay? Transcription is going to occur in the nucleus, whereas translation occurs in the cytoplasm. I want you to remember post-translational modifications. Post-translational modifications basically convert that newly synthesized protein to its final form. Okay? We're converting that newly synthesized protein to its final form. We also have a process of mRNA processing, right? One of those processes is called alternative splicing. And so the exons that encode for protein, they're good, but the introns are non-coding segments, so they're like spliced out. And so basically from alternative splicing, we can get one gene that can code for a few different products. Okay? So remember that we're going to be in a state of chemical disequilibrium, right? So when we're looking at the two fluid compartments, right, inside the cell and outside the cell, we're in a state of chemical disequilibrium. So that means that the chemicals are not going to be in equilibrium. They're not going to be in the same amount on the outside versus the inside of the cell. Like how we can see intracellularly, we have more potassium, right, and more proteins, right? However, in the outside, we have more sodium, right? More sodium on the outside. So when we look at sodium on the outside of the cell and look at sodium on the inside of the cell, it's disequilibrium. It's not equal, right? That's chemical disequilibrium. But we're in a state of osmotic equilibrium. So water essentially is the only molecule that can move freely between cells. And so we're in a state of osmotic equilibrium. Okay, same amount of water inside versus outside of the cell. We have different channel proteins, right, or different gated channels right? We have chemically gated channels, right? That's going to respond from like a chemical, like a ligand. We have voltage gated channels. Remember that voltage gated channels, they're going to open and close in response to a voltage change, right? The change in electricity. Okay. Then we have mechanically gated channels that are going to respond to like physical forces or increased temperature or pressure, things like that. Okay. That sodium potassium pump, right, is going to be one of the most important primary active transporters in the body. Okay. And we're moving a ratio, right? We're moving potassium into the cell while we're moving sodium out of the cell, right? Okay. But remember, this sodium potassium pump is going to be a important primary active transporter in the body. When we're looking at the nervous system, it's these neurons that with these processes that secrete these neurotransmitters. And this nervous system will exhibit emergent properties. And that's going to include consciousness, intelligence, and emotion, right? We are not able to tell what's going to happen from the individual connections, but it has these emergent properties, again, including consciousness, intelligence, and emotion. The central nervous system is going to consist of the brain and the spinal cord, whereas the peripheral nervous system is basically everything else. We also have the enteric nervous system, too. That's just simply a network of neurons in the walls of your digestive tract. Remember that neurons, they can't replicate themselves, right? Neurons can't replicate themselves because they don't have centrioles, right? Centrioles are those structures that are located outside the nucleus, important for the movement of nuclear DNA, okay? But again, neurons don't have centrioles, so once they die, they're just done, right? You can't replicate them. Nerves are just basically a bundle of axons, bundle in connective tissue, right? The epineurium, perineurium, and endoneurium, right? We have sensory nerves, motor nerves, and mixed nerves. The cell body is going to be the control center. The cell body is going to be the soma, right? That's going to contain that nucleus, okay? Off of that cell body, you're going to have these extensions right here. These are going to be those dendrites, right? These dendrites will receive incoming signals, okay? They receive incoming information. Okay? When we want to transport information or carry that outgoing signal, we're going to use an axon, right? These axons carry outgoing signals from this integrating center, which is the body, right, to that target cell, okay, through this axon. Okay? Dendrites receive information, axons carry out outgoing information. So remember the different glial cells, the neuroglia, right? And I'll talk about that in a second too. But remember also white matter. White matter is those myelinated areas, right? White matter is the axons. Okay. Gray matter is the cell bodies, right? The cell bodies are wherever there is communication. That communication basically is that synapsing. Okay. But again, white matter is the axons. Gray matter is the cell bodies, okay? When we're looking at the different glial cells, remember that we have glial cells of the central nervous system and glial cells of the peripheral. Peripheral nervous system glial cells are simply the Schwann cells, satellite cells. Schwann cells form the myelin sheath. Satellite cells support the cell bodies and the ganglia. The central nervous system, right, those neuroglia being the ependymal cells, astrocytes, microglia, and oligodendrocytes. Okay, remember which ones are where. Ependymal cells make up that cerebral spinal fluid. Astrocytes, right, they're going to make up that blood-brain barrier. Okay, blood-brain barrier. Okay, that we can see right there. Microglia, these are those, remember we talked about these are those modified scavengers, right? Those modified immune cells, right? Because remember, these are basically macrophages. Okay, these are those modified macrophages that's in the central nervous system. And then again, oligodendrocytes, myelin sheaths, right? So these are the four neuroglia of the central nervous system: ependymal cells, astrocytes, microglia, and oligodendrocytes. Okay. Also mentioning again that blood-brain barrier protects the brain, right? Those highly selective permeable capillaries are going to be basically covered, as you can see by these astrocytes. right? These astrocyte foot processes promote the tight junctions between the endothelial cells, right, of the blood vessels, not allowing anything to escape it. All living cells have a membrane potential. So the resting membrane potential is basically the difference in charge within that cell membrane, okay? And so the resting membrane potential is due mostly to potassium. And that's because the intracellular fluid has a higher concentration of potassium compared to the extracellular fluid, causing a net negative resting membrane potential, okay? Because of those leaky channels. When we're looking at this, remember the process of these membrane changes, right? So resting membrane potential is going to be around like 70 millivolts, right? Depolarization is when sodium rushes into the cell. Repolarization is when potassium rushes out of the cell. So if you look here, we're resting, it depolarizes, sodium enters, then it repolarizes, potassium rushes out, right? This stage of hyperpolarization is where that resting membrane potential becomes even more negative, right? And then it goes right back to its resting membrane potential. Remember, depolarization is when sodium rushes into the cell. Repolarization is when potassium rushes out of the cell. An action potential is important because it's basically an all or none principle. It will happen or it won't, right? An action potential occurs when a neuron sends information down its axon, right? Down its axon, away from the cell body. Okay, that's going to be triggered by that membrane depolarization. And again, when we're looking at it, sodium rushes in, then we get to a certain charge, those sodium channels close, those potassium channels open, and then potassium rushes out to repolarize the cell. Okay. Neurons, they're going to communicate at this synapse, right? We have electrical synapses and chemical synapses. When we have these chemical synapses, remember these chemical synapses are going to use neurotransmitters that are going to be housed in these vesicles, right? These terminal vesicles. And what happens is calcium binds to this vesicle and it induces the exocytosis of this vesicle. So this vesicle will spit out its compounds. Specifically, when we're looking at this right here, right, it's acetylcholine, right? Acetylcholine, the exocytosis of acetylcholine is induced by calcium. So say, for example, with that neuromuscular junction, right, calcium enters this axon terminal, tells this vesicle to release the acetylcholine into the synaptic cleft space. Now, the central nervous system had developed from a hollow tube. But before that hollow tube, we all began, right? Or the central nervous system all began as a neural plate, right? We started as this neural plate, and then it basically closed up, and then it became a neural tube, okay? It became a neural tube as it closed up, right? But again, started off as a neural plate, then it became a neural hollow tube. Okay. Central nervous system again consists of the brain and the spinal cord. Gray matter again is unmyelinated cell bodies, right, or a cluster of cell bodies, or the dendrites, axon terminals. This is where we have those synapses, right? This is all gray matter. So you can see right here in the spinal cord. White matter is the myelinated axons, right, or those axons that are traveling, right? So when we look at this, right, this is the axon portion, right? This is going to be white matter. Remember the different structures of the brain stem, right? So we have the medulla, right? The medulla is going to have those somatosensory tracts. It's going to have those pyramids. This is where we have that decussation or that crossing over. But also the medulla is going to have different centers, right? Cardiovascular, respiratory centers. The medulla is going to be able to control involuntary functions such as blood pressure, breathing, swallowing, vomiting, respiration, things like that. Okay. The pons also have some cardiac and urinary and respiratory functions as well. Okay. Coordinating the control of breathing, right? Your midbrain portion, right, is going to also have some eye movement, reflexes, and things like that. Okay. But remember that medulla for me, please. Remember the cerebellum. The cerebellum is going to be that little brain, but it coordinates movement. Coordinates movement. Okay, it's going to be able to process sensory information, coordinate and execute movement. Okay, unconscious. Okay, unconsciously. Okay, that's cerebellum. However, damage to this region, right, can lead to ataxia. Right, that's the impaired balance and coordination. Okay, can lead to ataxia. The thalamus, so now we're in the diencephalon region, right? That's going to contain those centers of homeostasis. The thalamus is going to be able to relay that information, that sensory information and motor information that's coming and going from that cerebral cortex, right? Remember, the thalamus is not going to be part of that limbic system. Okay, the thalamus is not a part of that limbic system. The hypothalamus is really important. We talk about that a lot. It's going to be the center for homeostasis, right? It's going to also be the center for behavioral states such as hunger, thirst, things like that. It's also going to influence autonomic function and endocrine function too, okay? When we look at the endocrine functions in the brain, right, we're going to have that hypothalamus, the pituitary gland, and the pineal gland. Pineal gland will produce melatonin, okay? Now, when we're looking at the limbic system, and I'll come back to the other ones for a second, but the limbic system is going to be the third region of the cerebrum, right? It's going to surround that brainstem, and it's going to contain places or structures such as the amygdala, which is going to be one of those almond-shaped structures, okay? And it's going to have a role in processing memory, decision-making, emotional responses, right? Including fear, anxiety, aggression, things like that. Part of this limbic system, we're going to have the cingulate gyrus, which is going to be linked to emotion and memory. The hippocampus is going to be associated with learning and memory, okay? Plays a vital role in regulating learning, memory encoding, memory consolidation, spatial navigation, right? Remember that, okay? The reticular activating system is going to control that of consciousness, right? Control wakefulness, sleep, muscle tone, and pain modulation. This is also going to be the site where general anesthetics act. Okay, this is going to be that reticular activating system, right? Again, controlling wakefulness, sleep, muscle tone, pain, consciousness. Okay. The hypothalamus does so much, right? But it's, again, we mentioned here that it controls two centers, feeding center and the satiety center, okay? Glycogen, when we're looking at energy, so energy is going to be stored in fat and glycogen. Glycogen is going to be glucose polymers, right? It's just going to be glucose, 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 right? A linkage of just glucose. And we store glycogen in the liver and in skeletal muscles, okay? Again, metabolism, I talked about it before, is going to be the sum of all chemical reactions in the body, right? Metabolism is basically the process of converting what you eat and drink into useful energy. Okay, we're able to extract that energy from nutrients, use it for practical work, chemical work, transport work, things like that, and store the excess. Okay, but again, it's going to be the process of converting what you eat and drink into useful energy. Metabolic pathways, we have anabolic pathways that build up larger molecules. We're synthesizing larger molecules from smaller ones. Anabolic pathways is going to be in the fed state. So when you just ate, you're going to build up larger molecules, right? Catabolic pathways break down larger molecules into smaller ones. So that's when you're in a fasted state. You haven't eaten for a while, you're going to start breaking down molecules, right? So you can start using them. When we're looking at these metabolic states, fed state, right? Again, it's simply the time following a meal. So you just ate. You're going to be using those products that you just digested, right? They were just being absorbed, and you're going to use them and store them in an anabolic fashion, right? You're going to be using them to build up more molecules. A fasted state, right? A fasted state is where the body has to tap into the storage energy reserves, right? You got to break down, for example, break down fat or break down glycogen, right? To get that energy out of it. Glycogen, right? Converts to glucose in this process of glycogenolysis to break down, to cut glycogen, right? That's in a fasted state. So you haven't eaten, you need to break down that glycogen to make glucose, put that glucose into your blood, right? Proteins can also be used to make ATP. I talked about that. Remember I said glucose, proteins, and lipids, they all can be used to make glucose, okay? Fatty acids, whenever we're trying to make energy or get energy from fatty acids, we undergo the process called beta oxidation. Beta oxidation will specifically produce acetyl-CoA. Too much acetyl-CoA or excess acetyl-CoA will become these ketone bodies, right? And these are going to be those strong metabolic acids that can lead to ketoacidosis, okay? The brain, focus on the brain here. The brain can use both glucose and ketones for energy. Can't use anything else, but it can use glucose and ketones for energy. The brain specifically is so hungry for glucose that it's going to use 50% of the body's glucose. Okay. Remember the pancreas. The pancreas is going to be this dual organ. It's going to have both exocrine and endocrine functions. Exocrine functions is from digestion, right? Endocrine functions is here. Because remember these cells, these islets of Langerhans cells that we can see. The beta cells produce insulin. Alpha cells produce glucagon. Remember that. Glucose metabolism is going to be controlled primarily by the ratio of insulin to glucagon, right? Gotta remember, in a fed state, insulin dominates. Insulin reduces blood glucose levels. You just ate. You're just bringing in sugar, right? We don't want that sugar to be in your blood. You want it to go to your cells, right? So that's what insulin is going to allow for that glucose to go into your cells. In a fasting state, when you haven't eaten, glucagon dominates. Glucagon raises blood sugar because you're in a fasted state. You haven't eaten, right? So your blood sugar is going to be low. So let's raise our blood glucose levels, okay? Because if your blood glucose levels are low, you're probably going to be not maybe confused, right? In a state of hypoglycemia. Diabetes mellitus is going to be a family of diseases, right? Diabetes mellitus is characterized by abnormally elevated plasma glucose concentrations. Type 1 is where you have insulin deficiency, like no insulin is being released because there's an autoimmune destruction of your beta cells. But type 2 diabetes is where you have insulin resistance, and this is going to account for 90% of all cases of diabetes. When we're looking at hormones, right, remember that the endocrine system is responsible for long-term ongoing functions in the body, right? Endocrine system is important for, these hormones are important for metabolism, regulation of internal environment, reproduction, growth, development. Remember those, okay? Also remember a simple endocrine reflex is going to include a stimulus that's going to be picked up by a sensor, right? That's going to have an input signal that's going to go toward the integrating center. That integration is going to happen. Then we're going to have an output signal that's going to go to one or more targets, and then we're going to have a response. Remember these steps. The hypothalamus, talked about it before, I'll mention it again, is going to be a gland in your brain that controls your hormones, right? Now, it's going to have neural function, where it's going to be able to control body temperature, hunger, thirst, circadian cycles, things like that. And it's going to have an endocrine function, where it's going to release these trophic hormones that act on the anterior pituitary, right? So I give you an example, right? We have TRH, thyrotropin releasing hormone that will go down this hypophyseal portal system to target those thyrotropes to release TSH, right? To then go to the thyroid gland, to produce T3, T4, right? Things like that, okay? The posterior lobe, right? So in the posterior lobe, we're able to store and then release two neurohormones. So we produce oxytocin and vasopressin, or also called ADH. We produce them in the hypothalamus up here. We just store them down here in the axon terminal. This axon terminal is in the posterior lobe of the pituitary gland. Okay, so again, oxytocin and vasopressin, or ADH, they are stored and then released from the posterior lobe. Okay. The anterior lobe secretes 6 hormones. Remember the 6 hormones: prolactin, thyrotropin, or thyroid stimulating hormone, adrenocorticotropin hormone, right? Growth hormone, follicle stimulating hormone, luteinizing hormone. Remember these 6 hormones. Okay? The pineal gland produces melatonin, right? Melatonin, we have the serotonin, actually. Serotonin is a precursor of melatonin that controls biological rhythms, body temperature, appetite, sleep cycles, things like that. Remember that, okay? Remember the adrenal cortex, right? The adrenal cortex secretes steroid hormones such as aldosterone, cortisol, those androgens such as DHEA. Okay? Aldosterone, right, is important in the RAS system, right? The renin-angiotensin aldosterone system. Aldosterone goes to the kidneys to reabsorb water and sodium, specifically sodium, and then water will follow sodium. Cortisol is going to be that main glucocorticoid, right? It mediates, remember. Cortisol mediates long-term stresses. It protects you against hypoglycemia, okay? Levels are naturally going to be higher in the morning, right? And diminish throughout the day, right? Cortisol is going to be high in the morning to deal with the stresses of the day, okay? And then those androgens, right? For example, DHEA or testosterone, things like that, those are going to be released by the zona reticularis. Again, these are steroid-based hormones. These are lipid-based hormones that can go easily through the cell membrane. Thyroid gland is going to synthesize these thyroid hormones. The synthesis of these thyroid hormones is going to take place in thyroid follicles. Okay, we need an enzyme, thyroid peroxidase. Thyroid peroxidase allows for these iodine groups to be thrown onto these amino acids, right? These tyrosine amino acids. Okay, that's why we're able to add this iodine group on there. Okay, thyroid peroxidase allows that. When T3 and T4 goes to their target cell, right? The target cell will use deiodinases. It'll take off an iodine from T4 to make it T3. T3 is the biologically active version. Okay, so that's what your cells want, T3. But the target cell will have an enzyme that will remove that iodine. Iodine is important to make these thyroid hormones, right? Iodine comes into your diet. You got to eat it, right? Iodine then gets into your cell via symport with sodium, right? Iodine gets into your cell via symport with sodium. When we're talking about calcium, right, calcium balance, thyroid, right, it produces T3, T4, but the thyroid gland also produces calcitonin. Calcitonin is a hormone that will reduce blood calcium levels, right? So we got to tone it down, right? Reduce blood calcium levels. On the opposite hand, parathyroid hormone increases blood calcium levels, okay? Because parathyroid hormone is going to take the calcium from your bones, right? It's going to tell your kidneys to don't pee it out, reabsorb it. And it's going to also tell your kidneys to convert vitamin D to calcitriol, the biologically active version of vitamin D, right? Vitamin D3. That vitamin D3 will then go to the intestines to absorb calcium from your diet so we can increase intestinal calcium. That's what the parathyroid hormone does. It tells your body, tells your kidneys to make calcitriol. Calcitriol then goes to the intestines, increase intestinal absorption of calcium. Opposite of calcitonin, because calcitonin wants to tone it down, right? Remember the other endocrine tissue, right? We talked about the pancreas, which is that dual organ that has both endocrine and exocrine functions. It produces insulin and glucagon, insulin from the beta cells, glucagon from the alpha cells, right? Basically to produce blood glucose levels. The kidneys also have endocrine tissue because they're going to produce EPO. EPO is erythropoietin. This will stimulate red blood cells, right, to be produced in the bone marrow. And this is in response to hypoxia, right? Low blood oxygen levels, right? Stimulate EPO, make more red blood cells, increase your oxygen carrying capacity, right? Kidneys also produce renin too. And I'll talk about that in a second. The thymus is a specialized organ that produces or releases substances to perform different functions in the body as well. Okay, and specifically it produces those thymosin hormones that direct the maturation of T cells. Now, the RAS system is the renin. So, RAS is the renin, angiotensin, aldosterone system, right? So if you look, change it to blue for a second, is RAS, right? Just put another A in there, right? That's the RAS system. So first, the kidneys produce renin, okay? This is going to be in response to low blood pressure. We want to raise blood pressure. So if you have low blood volume or low blood pressure, this system will kick in. Renin is released from the kidneys. So the kidneys, if we remember, produce 3 hormones. EPO, right, to make more red blood cells. Calcitriol, right, to increase calcium absorption. And now renin. Renin is released from the kidneys in response to low blood pressure. Renin will act with the already circulating angiotensinogen into forming angiotensin 1. Angiotensin 1 will then travel in the bloodstream, okay, to go to the lungs, where you're going to find an abundance of ACE enzyme. ACE stands for angiotensin converting enzyme. All right? Angiotensin converting enzyme will convert angiotensin 1 into angiotensin 2. Angiotensin 2 is going to do everything it possibly can to raise your blood pressure. So it's going to be a powerful vasoconstrictor. So it's going to constrict your blood vessels, right, to raise your blood pressure, number one. Okay. It'll go to your hypothalamus. It'll tell your hypothalamus to release ADH. So we can, again, reabsorb fluid, water. Angiotensin 2 will also go to your adrenal cortex. Tell your adrenal cortex to release aldosterone. Aldosterone then goes to your kidneys to reabsorb sodium and water, thus increasing your blood volume, thus increasing your blood pressure. Okay, so this, in a nutshell, is the renin, angiotensin, aldosterone system, and we'll talk about that in much more detail too. Okay. There's going to be general properties of the sensory system, right? There's going to be 4 major groups of sensory receptors: chemical, mechanical, thermal, and photo. There's going to be 5 taste or gustation sensations, not 4. We have sweet taste buds, sour, salty, bitter, and umami. Also, remember the type of sensory receptors, right? Chemical receptors or chemoreceptors, mechanical receptors, photoreceptors, and thermoreceptors. When we're looking at other sensors, right, visual, right, is the sense of light, right? So light is going to be reflected off from objects in the environment. They're going to be translated into a mental image. Is these retinal cells that include rods, bipolar, cones, horizontal, and achromal cells, right? When we're looking at our hearing apparatuses, right, or apparatus, the inner ear will consist of our equilibrium apparatus, which is going to be those semicircular canals. The semicircular canals, they're going to have cristae in them, right? And that's going to be able to monitor rotational acceleration of the head. For hearing is going to be the cochlea, beginning at the oval window, right? That oval and round window. It's going to have stereocilia in there that's going to contain ion channels that will transduce vibrations, right? And this is how we're able to pick up sound sensation. Olfaction is going to be the sense of smell. Remember the olfactory nerve, that olfactory cranial nerve number 1, olfactory nerve? It does not pass through the thalamus for integration, okay? Nociceptors will initiate protective responses. These nociceptors, these are basically free nerve endings found in your skin, variety of organs in your GI tract, but these are pain receptors. These are pain receptors, and remember that referred pain is going to be poorly localized pain perceptions from visceral and somatic pain pathways converging on a single ascending tract, so you'll feel it somewhere else, basically, okay? When we talk about blood, right, blood is going to be composed of plasma and formed elements. Plasma is going to be the extracellular matrix. That's going to be the fluid, right, consistent of water. In that water, you're going to have proteins and ions and gases, trace elements, vitamins, things like that. Those proteins being the albumins, the globulins, the fibrinogens, the transferrins. Remember those plasma proteins, okay? The formed elements, right? Formed elements, right? These are going to be right here. These are going to be the cellular components, right? Those cellular components being the red blood cells or the erythrocytes. These are going to be carry and transport oxygen and carbon dioxide. White blood cells. These are going to be those leukocytes, lymphocytes, monocytes, neutrophils, eosinophils, and basophils. Important in your immune system, right? And then the other formed element being the platelets. These are going to be thrombocytes. They're going to split off from that grandmother megakaryocyte, and they're going to be important for blood clotting. Okay? Remember these plasma proteins that I mentioned. Most of them are going to be synthesized by the liver. Right? You're not going to have those antibodies synthesized by the liver because they're going to be synthesized by those B cells. But all those other plasma proteins, they're going to be made by the liver, the albumins. Smallest, but they are important because they contribute to the osmotic pressure. Okay, very, very important. Globulins, these are transport proteins. They can transport ions and things like that, or they can be immunoglobulins or antibodies that are going to attack pathogens. And then these fibrinogens are going to be the largest, but they're going to be involved in blood clotting processes. Okay, remember these. Blood cells come from bone marrow, right? Through a process called hematopoiesis, right? This is how we produce bone cells or blood cells, right? Hematopoiesis is going to be controlled by cytokines. Cytokines are basically peptides or proteins that are released from one cell that affect the growth or activity of another cell. So we have erythropoietin, like I told you. Erythropoietin is produced by the kidneys to stimulate red blood cell synthesis. Okay. Thrombopoietin is going to stimulate thrombocyte platelet production, right? And again, erythropoietin was stimulated by a process called erythropoiesis, stimulates EPO, erythropoietin, again, from the kidneys, targeting red blood cell synthesis. Hematocrit is just simply the ratio of red blood cells in your blood, okay? It tells you the ratio of red blood cells to your plasma, and it tells you about your oxygen carrying capacity of your blood, okay? Red blood cells, right? So specifically mature red blood cells lack a nucleus. And remember the morphology of these red blood cells, right? The morphology can provide clues to the presence of a disease, okay? Aged, I gotta let you know, aged and damaged erythrocytes are gonna be broken down by the spleen, right? Under normal conditions, right? Old and damaged red blood cells, they're gonna go to the spleen to be recycled, okay? Again, they're gonna be phagocytized by the spleen, okay? This spleen right there. Hemoglobin is going to be very important. Hemoglobin is a molecule that helps to transport oxygen, right? Hemoglobin is going to be made up of 4 globular protein chains, right? 2 alpha, 2 beta. And because we have 4 protein chains, we're going to have 4 heme groups, and we're going to have 4 iron molecules in each heme group, okay? Each iron is able to transport 1 oxygen, right? So we're able to transport 4 oxygens per hemoglobin molecule, okay? Hemoglobin's affinity for oxygen is really regulated by how easily hemoglobin acquires and releases oxygen molecules in that fluid that surrounds it, okay? That's its affinity. How likely is it to grab and release it? Hemostasis simply prevents blood loss from damaged vessels. Hemostasis will begin when you have damage to the wall of your blood vessels, and it's that exposed collagen. That exposed collagen starts this process, right? It starts these platelets. These platelets will adhere to that collagen wall. These platelets will then release platelet factors, and this positive feedback loop where it will attract more platelets and more platelets and more platelets, and these platelets will aggregate, right? And basically form this platelet plug, right? This platelet plug then gets reinforced by a clot, okay? But again, it's going to start with this collagen being exposed, okay? When we're trying to, when we're looking at, sorry, when we're looking at this coagulation cascade, right? This coagulation converts this platelet plug into a clot, a more stronger clot. We have an intrinsic pathway, right? We have two pathways, intrinsic and extrinsic pathway. The intrinsic pathway is known as that contact activation. Again, it's going to be activated by that exposed collagen. Okay. The common pathway, remember the common pathway is going to begin with factor 10. Factor 10, also factor X, right? 10, right? It's just X for 10 for Roman numeral. And remember alveoli. Alveoli are going to be the sites of gas exchange. Remember type 1 cells are going to be thin, allowing for gas exchange. Remember these type 2 cells, right? They're going to secrete surfactant, right? Preventing your alveoli from collapsing. And of course, we have alveolar macrophages that's going to be there to scavenge any type of debris or pathogens. Remember the lungs, right? The lungs work in coordination with the muscles, right? It's the muscles, the diaphragm, and the external intercostals. When they contract, that allows for more air to come into your lungs, right? And then simply expiration is when those muscles recoil, they relax. Remember this nerve innervation. The phrenic nerve causes the diaphragm and the respiratory muscles to contract, whereas the vagus nerve will regulate respiratory rate, okay? And finally, I'll end with obstructive lung diseases. Obstructive lung diseases is going to be difficulties with inhalation, right? So you have increased airway resistance, Obstructive lung diseases, you can't breathe out. Things such as asthma, COPD, obstructive sleep apnea. These are cases of obstructive lung diseases, okay? That's it. Happy studies. This was your midterm lecture review.